Now we will explain amino acid metabolism that consists of catabolism of amino acid and biosynthesis of amino acid. Amino acids are not stored by the body and must be obtained from the diet because some amino acids cannot be synthesized by human body, called essential amino acid, and amino acids synthesized de novo or produced from normal protein degradation. As well, any excess amino acid biosynthesis of the cell rapidly degraded. Amino acid catabolism, there are two phases for amino acid degradation. The first phase is removal of alpha amino group by transamination and subsequent oxidative deamination to form of alpha keto acid and ammonia. Most of ammonia is used in the synthesis of urea and the remainder is excreted in urine. The second phase, amino acid will be converted to alpha keto acid and then the carbon skeleton of alpha keto acid converted to common intermediate of energy producing, and this intermediate of metabolism is used in glucose synthesis or fatty acid ketone body glycogen synthesis, which are catabolized to yield of CO2 and H2O and energy. Amino acid pool. There are three sources of supplied amino acid pool. There are three sources of supplied of amino acid pool. The first sources is degradation of body protein in about 400 grams per day. The other sources is amino acid results from dietary protein or amino acid that synthesis in human body and called non-essential amino acid. Amino sources that depletion of amino acid pool. The first one is synthesis of body protein in about 400 grams per day. So the amount of protein synthesis and protein degradation is equal. This is called protein turnover, which means the amount of protein degradation is equal to the amount of protein synthesis. The second is synthesis of some nitrogenous compound from amino acid, which results to depletion of amino acid pool. Like porphyrin, creatine, neurotransmitters compound, purine, pyrimidine, etc. And the last one results from the degradation of catabolism of amino acid to yield of end product that's used in the synthesis of glucose, glycogen, ketone body, fatty acid, steroid, or can be enter Krebs cycle to form of CO2 and H2O and energy. Catabolism of amino acid involve removal of amino group followed by breakdown of the carbon skeleton to produce 7 intermediate products: pyruvate, fumarate, acetyl coenzyme A, acetoacetate, oxalate, alpha-ketoglutarate, and succinyl coenzyme A. Thus, can be entered directly in the synthesis of glucose, lipid, glycogen, ketone body, or oxidation to production of energy and converted to CO2 and H2O by Krebs cycle. You know, a Krebs cycle, each cycle of a Krebs cycle yield 12 of ATP units. Amino acid can be classified to a glucogenic and ketogenic. or ketogenic and glucogenic. You know, amino acid can be classified to according to the polarity, polar and non-polar, or according to the net charge, positive, negative, or neutral charge, or can be classified to the essential and non-essential amino acid. Non-essential, that means amino acid which can be synthesized by the body, and essential amino acid, this type of amino acid cannot synthesize by the body and must be obtained from the diet. Amino acid classified to the glucogenic or glucogenic and ketogenic or ketogenic depend on which of the 7 intermediate are produced. This classified is depend on which of the 7 intermediate are produced. So, a glucogenic acid, the end product of catabolism, this type of amino acid is pyruvate or one of the intermediate of a Krebs cycle. And this, the end product is used as substrate for gluconeogenesis, so an increased level of glucose in liver and kidney. This type of amino acid. While ketogenic amino acid, leucine and lysine, are the only ketogenic amino acid found in protein. And the end product of catabolism of this amino acid are acetoacetate or one of its precursors like acetyl coenzyme A or acetoacetyl coenzyme A. And the carbon skeleton of this amino acid is not used in gluconeogenesis and so is not an increased level of glucose. Now, amino acid that form oxaloacetate, like asparagine. Asparagine is hydrolyzed to form of aspartate and liberate ammonia, and this reaction is required of asparaginase. Then aspartate is converted to oxaloacetate by a transamination reaction, and alpha-ketoglutarate act as acceptor for amino group from aspartate to form a glutamate, and this reaction is required aminotransferase. B. Amino acid that form alpha-ketoglutarate, like amino acid that form alpha-ketoglutarate via glutamate are glutamine, proline, arginine, histidine. A glutamine is converted to glutamate by removal of amino group and in the presence of glutaminase enzyme. Glutamine is a glucogenic and non-essential amino acid. Then glutamate is converted to alpha-ketoglutarate by oxidative deamination in the presence of glutamate dehydrogenase enzyme, or converting to alpha-ketoglutarate by a transamination reaction. 2. Proline. Proline is a glucogenic acid and non-essential, is oxidized to form of glutamate, and then glutamate will be transaminated to alpha-ketoglutarate or oxidative deaminated in the presence of glutamate dehydrogenase enzyme to form of alpha-ketoglutarate. 3. Arginine. Arginine is cleaved to ornithine in the presence of arginase enzyme, and this type of reaction is occur in liver as part of urea cycle. Arginine is a glucogenic and non-essential amino acid. Then ornithine converted to alpha-ketoglutarate. 4. Histidine. Histidine is by oxidative deamination, the presence of histidase enzyme, liberate of ammonia and form of urocanic acid. The urocanic acid is converted to N-formimino-glutamate, and then in the presence of tetrahydrofolate, transport formimino group to tetrahydrofolate and form of a glutamate, which converted to alpha-ketoglutarate. Glutamate can converted to alpha-ketoglutarate by transamination or by oxidative deamination in the presence of glutamate dehydrogenase to form of alpha-ketoglutarate. Amino acid form pyruvate. Amino acid that form pyruvate as the end product of catabolism are alanine, serine, glycine, cysteine, threonine. Alanine will be converted to pyruvate in the presence of alanine amino transferase enzyme. This is a reversible transamination. This reaction is reversible. And alpha-ketoglutarate act as ammonia group acceptor to form of glutamate and give pyruvate. Alanine is a glucogenic and non-essential amino acid. The second one is serine. Serine is converted to glycine by removal of methylene group from serine to tetrahydrofolate to form of glycine. Serine is a glucogenic and non-essential amino acid. A glycine, the third amino acid, is a glycine. Glycine is converted to serine. Glycine is converted to serine by reversible addition of methylene group, methylene group from N5 and 10 methylene tetrahydrofolate, which act as one carbon acceptor. This is the coenzyme to give off serine. Then serine will be hydrolyzed to form of pyruvate and liberate of ammonia group. Dehydration, sorry. Or glycine can be oxidized to form CO2 and ammonia. Or glycine can be converted to a glycosylate, which oxidized to oxalate, and by transaminated to a glycine. Oxalate transaminated to a glycine in the presence of transaminase enzyme. Any deficiency in a transaminase enzyme could lead to overproduction of oxaloacetate and kidney damage. And this type of disease called primary oxaluria type 1. Cysteine. For cysteine amino acid is results from condensation 2 molecules of cysteine, which form a thiol bridge to form of cysteine amino acid. So when reduced cysteine will be give 2 molecules of cysteine, and this reaction require NADH and H positive charge, which act as reductant, and then cysteine will be desulfuration to form of pyruvate. The 5th amino acid is threonine, is a glucogenic and essential amino acid, so it's converted directly to pyruvate or converted to alpha-ketobutyrate, which form converted to succinyl coenzyme A. Amino acids that form fumarate are phenylalanine and tyrosine. Phenylalanine and tyrosine. Phenylalanine is essential while tyrosine are non-essential amino acid. Both amino acids are glucogenic and ketogenic amino acid. So phenylalanine is hydroxylation to form of tyrosine. And this reaction is require tetrahydrobiopterin and molecular oxygen as coenzyme for hydroxylase enzyme to be active. Molecular oxygen, one atom of molecular oxygen is used in form of hydroxyl group of tyrosine, while the other atom is used in form of water. And tetrahydrobiopterin can be regeneration from dihydrobiopterin when reducing. So tyrosine then can be converted to fumarate and acetoacetate. Amino acid form succinyl coenzyme A. Amino acid form succinyl coenzyme A. Methionine is one of this amino acid, and amino acid will be converted to S-adenosyl methionine, and this reaction is require ATP to hydrolyze for 3 inorganic phosphates. S-adenosyl methionine is high energy and represent the major methyl group donor. So this compound is converted to S-adenosyl homocysteine, then hydrolyzed to give adenosine and homocysteine. Homocysteine condense with serine to form of cystathionine, and this reaction is require vitamin 6 water-soluble vitamin or pyridoxal phosphate. And cystathionine then converted to cysteine and alpha-ketobutyrate. Alpha-ketobutyrate will be decarboxylation to form of propionyl coenzyme A and then converted to succinyl coenzyme A. Methionine can be re-synthesis from homocysteine, and this reaction requires tetrahydrofolate as methyl group donor to give methionine and tetrahydrofolate. The second, the other amino acid that form succinyl coenzyme A are valine, isoleucine, and threonine. Valine and isoleucine will be decarboxylation to form of a propionyl coenzyme A and then to succinyl coenzyme A, and to convert propionyl coenzyme into succinyl coenzyme A require biotin and vitamin B12. And the propionyl coenzyme A can be regeneration by some amino acid and catabolism odd number of free fatty acid. Propionyl then can be converted to succinyl coenzyme, as well as threonine. Threonine is dehydration to give alpha-ketobutyrate and then decarboxylation to give a propionyl coenzyme A and then to the succinyl coenzyme A. Or threonine can be converted to pyruvate. Threonine is a glucogenic and essential amino acid. Amino acid form acetyl coenzyme A or acetoacetyl coenzyme A. There are 6 amino acids that form acetyl coenzyme A or acetoacetyl coenzyme A: leucine, isoleucine, lysine, tryptophan, phenylalanine, and tyrosine. Leucine converted to acetoacetyl coenzyme A and acetyl coenzyme A. This amino acid is ketogenic and is essential amino acid. Isoleucine converted to acetyl coenzyme A and propionyl coenzyme A, which then converted to methylmalonyl coenzyme A, and the last end product is succinyl coenzyme A, which enter Krebs cycle. Isoleucine is glucogenic and ketogenic and is essential amino acid. Lysine is ultimately converted to acetoacetyl coenzyme A. Tryptophan is converted to alanine and acetoacetyl coenzyme A. Then alanine will be transaminated to pyruvate. Tryptophan is glucogenic and ketogenic, amino acid. Phenylalanine and tyrosine. Phenylalanine will be hydroxylation to form of tyrosine, and then tyrosine will be converted to acetoacetate and fumarate. Now, catabolism of branch chain of amino acid. Isoleucine, valine, leucine. This is our essential amino acid and our branch chain amino acid. This reaction will be, this 3 amino acid will be converted to alpha keto acid by removal of amino group by transamination reaction and in the presence of amino transferase enzyme. This reaction requires vitamin B6. And then alpha keto acid by oxidative decarboxylation, removal of carboxyl group from alpha keto acid to form of Barry compound. And then dehydration to form or different end product, acetyl coenzyme A, succinyl coenzyme A, acetoacetate, and acetyl coenzyme A. And this branch chain amino acid, in contrast to other amino acids, they are metabolized by peripheral tissue like muscle rather than liver. So the 3 amino acid have the same rate of catabolism. Thank you.